Key considerations for accurate photoluminescence measurements. Photoluminescence measurements are crucial in material science and semiconductor research, providing insight into the electronic and optical properties of materials. Accurate measurement require attention to factors such as sample preparation, experimental setup, and data analysis. Important considerations include the choice of excitation source, detection equipment, and the environmental conditions, as well as the calibration and alignment of optical components. Properly addressing these aspects ensures reliable and reproducible results for meaningful conclusions in PL studies. In this video, I will talk about the 6 important points to remember while measuring the photoluminescence. For solid samples, PL measurements are more sensitive to minor defects or structural changes compared to the absorbance measurements. For example, introducing trap states in a semiconductor does not induce much changes in the absorption spectra, whereas it can quench the PL completely. Intensity lowering in PL is called quenching. Thus, PL is more sensitive to minor defects or structural changes than absorption spectra. Second point is increase in the integration time to increase the PL intensity. You can increase the integration time of your measurement in order to increase the signal intensity. Integration time can be adjusted by the software which using for PL measurement. The dotted line represents the PL with the higher integration time than the solid one. However, make sure that the highest signal should be below the saturation level of the detector. Otherwise, it is harmful for the detector, and also if it reaches the saturation point, the spectra will look like this, flat at the peak maxima. Third point is reduce signal to noise ratio by averaging the spectra. If your signal to noise ratio is higher for your sample, then you can increase the number of averaging the spectra. This is known as spectral averaging. For example, if this spectra took with an averaging of 2, and if we increase the number of averaging to 5, the resultant spectra will be look like this. Thus, by doing spectral averaging, we can reduce the signal to noise ratio of your PL spectra. Fourth point is adjusting the gain to amplify the signal. If your PL spectra is particularly very weak, then you can adjust the gain to amplify the signal. For example, first one without amplifying and the second one is with amplifying. Did you notice anything other than the sample intensity increases? Yes, noise level also increases. Thus, always keep in mind that amplifying the signal will amplify the noise level also. Coming to the fifth point, maintaining the experimental setup, same for multiple sample measurement. Several factors affect the PL intensity. If you change any of them during the measurement, your data may not be comparable. Next, we can have a look on the factors affecting the PL intensity. That is, first is source intensity or excitation intensity, excitation wavelength, sample orientation, and the detector angle and the distance between the excitation source, sample, and the detector. The first one is source intensity or excitation intensity. Intensity of source depends the intensity of PL. That is, the source intensity higher means higher the PL intensity. Here is an example of PL intensity with respect to the excitation intensity. First one with low excitation intensity and hence low PL intensity. Second one is higher excitation intensity and hence higher PL intensity. Second one is source wavelength. Depends on the excitation wavelength, different intensity of PL will results. Sometimes the nature of the PL spectra will change depends on the excitation wavelength. Here is an example of two different excitation wavelength, yellow and green. Resultant PL intensity also shown here. Some material will exhibit this property and that is the excitation dependent PL spectra. Third point is sample orientation. Normally, the excitation and emission monochromators are arranged in particular way, that is perpendicular to each other. The angle between excitation and emission monochromator is 90 degree. Sample orientation influences the PL intensity. Here shows an example. First case, the sample is oriented in this way and the PL intensity is higher. Second case, the sample is placed in this way and it shows a reduced PL intensity. The best sample position is 45 degree angle. That is, the excitation to emission monochromator is 90 degree, and in this 90 degree angle around 45 degree is the best position for sample placement. Next is detector angle and the distance between the excitation source, sample and the detector. This is the distance between the excitation source and sample. These distance and detector angle also influence the PL intensity. So for multiple measurement, don't change anything if you want to compare the data. Sixth point is the usage of integration sphere to find out the PL intensity. What is an integrating sphere? An integrating sphere is an optical device used to measure the total light from a source by capturing and integrating light within a hollow spherical cavity with a highly reflective interior surface. It evenly distributes light, allowing for accurate measurements of diffuse reflectance and transmittance, as well as the total luminescent flux of a light source. BaSO4, barium sulfide, is commonly used as a coating material for the interior of the integrating sphere due to its high reflectance and diffuse scattering properties across a broad range of wavelength. This ensures minimal absorption and consistent light distribution within the sphere, leading to the precise and reliable measurement. Without integrating sphere, your PL intensity will not be reliable. However, you can compare PL intensities between samples if you kept all the other factors same. Alternatively, for comparing the data, you can normalize the spectra to highlight the changes in PL maxima or peak shape, whether sharpening or broadening, and the PL wavelength range, etc. Thus, the key considerations for the accurate photoluminescence measurements are the following points. First one, the PL measurements are more sensitive to minor defects or changes in structure. Second one is the integration time to increase the intensity of the PL signal. Third one is the spectral averaging to reduce the signal to noise ratio. And the fourth point is adjust the gain to amplify the PL signal from your sample. And the fifth point is if you are measuring multiple samples, ensure that your setup stays same throughout. And the sixth point is integrating sphere is usable for PL intensities. If you think the video is useful, please like, comment and subscribe our channel. Thank you for watching.